Yeah, I mean, there's no way to do the video here. It's just, but I got the screen shared on there. All right. So we're probably not going to get everything done today that I have on this a little bit. So, but I think we'll get close. So whatever we don't get done, I'll try to just pick up the next lecture. We do have a practice quiz for you today. It does not count for your grade. I'm not going to even record the grades. I asked you to bring a Scantron in, just so that I can maybe get an average and a deviation. Just curious. You don't have to. So we're going to do that at the end though, if that's all right. So I got an alarm set here for 11:30. And there's no quiz today, but I can tell you there's a pretty good chance we might have one next week. So remember, those quizzes are just 5 questions. We'll take it first thing that we do that day. I'm just going to put up 5 true false statements on the projector, and you don't need a Scantron or anything for that. You just write T or F on a piece of paper with your name. Okay, we're going to have 6 of those. The lowest grade will get dropped. I think it's 6. All right. Any major questions about anything from last time? All right. So this is still all pretty introductory, and it should be review for anyone that's taken Bio 1. I think we left off last time talking about proteins. And so now we're going to get into a little more specifics about proteins. Remember, proteins are the functional molecules of the cell, right? The central dogma dictates that the cell moves information in the form of DNA to the intermediate molecule RNA, and then ultimately towards the functional molecule proteins. There's a lot of different classes of proteins. In general, you could think of them as being in just 2, although there's certainly more subdivisions with those 2, but you could think of them as being structural and functional. Right? And the functional proteins are these guys, the enzymes. Right? Enzymes are proteins that catalyze chemical reactions. What does that mean if you catalyze something? You speed up a chemical reaction. So in order to have life, I told you guys last time, what are the 2 things that you need to do to have life? Complexity, diversity, and then here's another one, speed. All right, you have to be able to do chemical reactions a lot more quickly than they could perform on their own, like millions of times more quickly. And that's what these enzymes are capable of doing. They're capable of speeding up reactions almost exponentially. All right, so the way that enzymes work is they lower this value called EA, which is your activation energy. When you look at a typical chemical reaction, right, you're going from substrate to product, right? So the substrates bind to the enzyme. The enzyme essentially positions the substrates in the correct conformation. We call that a transition state. And then whatever catalytic activity needs to be performed gets performed. Having the substrates in that transition state is the primary event that reduces the activation energy. So you can think of activation energy as like a hurdle that a reaction has to get over in order to complete, right? So you're going from substrate to product, and in order to do that, you have to get over this hump, right? So a catalytic enzyme comes in and just lowers that hump, right? It has a lot to do also with Gibbs free energy. It's basically, negating your value. It's bringing it down more so that it gets closer to occurring spontaneously. All right, so enzymes, it's not like they just do their job and then they're kaput, right? So they are recycled. They can work over and over again. Enzymes will bind substrates, they'll initiate the reaction, and then they'll release the substrates and they can be recycled. All right, so how well enzymes work, in vitro are dictated by the conditions that they are in. So typically we look at temperature, it's a big one. We're going to do a lab exercise on this. I think maybe it's our first one. No, it's our second one. So we look at temperature, we can look at pH and also ionic strength, right? So the environment, the salt environment, the salt aqueous environment that the enzyme is in is going to dictate how well it works as well. The binding of the substrate to the enzyme occurs by all these non-covalent interactions, hydrogen bonds, ionic bonds, and also hydrophobic and hydrophilic interactions. Enzymes are very specific for the substrates that they work on. This is frequently termed the lock and key model of molecular biology, which means, you got one lock that works for one key that works for a lock. You have one enzyme that works for a specific substrate. So here we've got our enzyme. It's binding substrates at the active site. The active site is also the catalytic site. And, it positions the substrates in the correct conformation that, is going to require the least amount of energy for the reaction to occur. So here we're taking a blue thing and a red thing and we're covalently sticking them together. It's a good example of like a polymerization reaction if you're building carbohydrates or proteins or nucleic acids. Think of these substrates as just like in random entropy disorder, right? They're floating around in solution. In order to position them correctly, that's going to be a random event, right? That would require a lot of probability and a lot of energy to do that. That's why these enzymes, because they can put it in a correct transition state, lower that activation energy. So that's the key event, the transition state. All right, after you get the initiation of transition state, whatever catalytic activity needs to occur occurs. But that transition state is key. That's what lowers the activation energy. All right, we also call, so oftentimes the enzyme will undergo what's called a conformational change. Whenever you hear that term, think shape change, right? And shape change in biology is essential. Remember, structure determines function. So if the shape of something changes, so can the function. All right, here's two examples of enzymes, chymotrypsin and trypsin. What is trypsin? You guys remember from A and P? We got some A and P people in there. Trypsin is a proteolytic enzyme. And what does that mean? Proteolytic. Protein, lytic, break. It cleaves proteins, it cuts proteins. Chymotrypsin and trypsin are two proteases, right? So they break up proteins. We have them in our in our GI tract, right? They're part of the enzymes that break down proteins and brought into our diet. So these are specifically their serine proteases, right? Which means that it's a serine residue on a protein, which is where they're going to cleave. Okay. So, the shape of the enzyme here enables the protein to correctly dock in. Right here, you can see you've got this ring structure, which are very hydrophobic, right? So it can fit into this pocket right here. And here we have an ionic reaction. So this residue right here is a positive charge. It can bind into this pocket because of the negatively charged aspartic acid. All right, so an important component of enzymes is also non-protein molecules that assist the enzymes. We call these coenzymes. Okay, so coenzymes are non-protein molecules that help the enzyme function. We've learned about a couple already. Can anyone think of what might act as a coenzyme? So we talked about ATP, right? The energy currency of a cell. That's certainly an example of a coenzyme. Another big one is this chemical that has a big name. I don't have it up there. So nicotinamide, adenine,
dinucleotide of NADH.
So nicotinamide adenine dinucleotide is a electron carrier, right? So the ability to carry electrons and also the protons that go along with them is an important feature in chemical reactions because it can provide those electrons at a higher energy state to the chemical reaction, right? So NAD plus and its cohort, NADH, right, are important electron carriers in specifically oxidation and reduction reactions, right? So remember from chemistry, oxidation is a loss of an electron, reduction is a gain of electron. That does seem counterintuitive since the word reduction is to lose. Sorry, I'm moving this thing. NADH can readily lose and gain an electron, and it can transfer that to another molecule. So it's enhancing a reaction, and that's why we call these coenzymes. Another component of enzymes are these prosthetic groups. So these are all these things are non-protein, right? But prosthetic groups are usually like oftentimes inorganic molecules like iron, zinc, things like that that are necessary to assist enzymes. DNA polymerase, right? The enzyme that catalyzes the addition of DNTPs to a growing strand of DNA. It requires magnesium to function. So whenever you're doing in vitro PCR, you have to include magnesium as a prosthetic group cofactor in that reaction. So often these chemicals are derived from vitamins. So animals have evolved to not produce some of these chemicals on their own. Some of these we have to get in our diet. And for example, niacin is used as a preliminary molecule to make NAD. Riboflavin makes another electron carrier called flavin adenine dinucleotide. So these NADH and FAD are really prominent in the electron transport chain and glycolysis. And when we talk about that later on, you're going to see them come up. All right, so here's NADH and NAD plus undergoing oxidation reduction reactions. So NAD plus is there in A, right? So when it gains an electron, it becomes NADH, right? So is that oxidation or reduction? Going from left to right. Reduction, right? Going from right to left is oxidation. All right, so a lot of factors regulate enzyme activity. We talked about some of them, such as pH, ionic strength, and temperature. Right. So, whenever you deviate from the isoelectric point of a protein enzyme, right, you're going to change conformation of it. So, but there are other ways of regulating enzyme activity too, which involve molecules. So, and this is important that, the enzymes are not working when they don't need to, right? And that they are ensuring that they are working when they do need to. So one way of regulating enzyme activity is by competitively inhibiting it, right? So basically you have a molecule that looks like the substrate, but is not. So it will bind to the enzyme. And so now the enzyme is wasting its time working on that competitive inhibitor instead of the actual substrate. When you go to the pharmacy, a majority of those compounds in there are competitive inhibitors of enzymes. The other way of inhibiting an enzyme is working on a site of the enzyme called its allosteric site. Okay, so you have the active site of an enzyme. Enzyme is the active site. But let's say a site on the enzyme has been recognized that if you stick something onto it, you end up changing the conformation of the enzyme and now it doesn't work. We call these allosteric sites. This is what a lot of protein biologists get right and talk at the same time. A lot of protein biologists are trying to recognize so that they can, or identify so that they can look at new drug targets. All right, enzyme activity has pathways, right? So from start to finish, and oftentimes it's a series of reactions that occur from different enzymes in a particular biochemical pathway. In order to regulate that pathway, something like feedback inhibition can occur. So let's jump ahead here. There's a good example of the reaction to produce isoleucine. So threonine is modified by threonine deaminase, and it ends up making an intermediate molecule called alpha-ketoglutarate. Right? Alpha-ketoglutarate then goes through a series of reactions to become isoleucine. Right? So what is threonine and isoleucine? What are these molecules? These are amino acids, right? So that's how you make isoleucine. But the more isoleucine you have, right, it will eventually start to inhibit threonine deaminase. So you will inhibit this pathway. So you're not the cell is not wasting energy making isoleucine if it has a lot. So this is called a negative feedback loop. And a good example of regulating enzyme activity. All right, so this figure, which I skipped over real quick, goes over regulating enzyme activity as well through either competitive inhibition or allosteric inhibition. Okay, so if you look up here, this is this figure. So here we've got the enzyme is active. It's got a substrate bound to the active site. But if we flood the system with a competitive inhibitor, which will bind to the active site but can't undergo a chemical reaction, you can see the competitive inhibitor is bound, but the substrate cannot actually bind. Okay. The other way of regulating an enzyme would be to latch on to its allosteric site with some sort of chemical. And when you do that, you can see it changes the conformation of the enzyme, it changes the shape of it, and now the substrate can't bind to it. Okay, so those are two prominent ways of regulating enzyme activity. Another way of regulating enzyme activity is through the addition of phosphate groups. The phosphorylation is a very key event in cell biology. It mediates a lot of cell signaling pathways. Think of it like a molecular switch that turns proteins on or off. Oftentimes, if they're phosphorylated, they're in their on position. And when they're dephosphorylated, they're in their off position. It's not always true. Sometimes it's reversed, but usually it is. Additionally, this is a relatively easy thing to pick up in the lab. Like if you want to see if a protein is phosphorylated in your pathway, there's a simple procedure called a Western blot, which we're going to talk about. And we're going to do a Western blot in the lab in this class, which uses antibodies to probe for proteins. And they actually have alternate forms of these antibodies that look specifically at the phosphorylated form of these proteins as well. But phosphorylation is just the addition of a phosphate group to a protein. There are 3 amino acids that are readily phosphorylated on proteins, and those are serine, threonine, and tyrosine. Definitely need to memorize that. All right, so those are the residues. Those are the amino acids on a protein that can be phosphorylated. The enzymes that do the phosphorylating are called kinases. The enzymes that do the dephosphorylating, moving the phosphates, are called phosphatases. Okay, so phosphorylation is mediated through the activity of kinases and phosphatases. And they are transferring inorganic phosphate. Now, where do you think it's getting the phosphate from? Very good. Spoiler up there. All right, so here you can see you've got an inactive enzyme, glycogen phosphorylase. Okay. And this phosphorylase kinase, right, phosphorylates the enzyme And when you add phosphate groups on the enzyme, you expose the active sites. So now it's in its on position. All right, substrates can bind to that enzyme. It's now working. Alternatively, the phosphatase now can come along and remove those phosphates as well. Okay, the phosphate is being acquired from ATP, right? So here you've got two molecules of ATP that are each losing one phosphate to provide this reaction. So when you move a phosphate, not only is there some energy transfer involved because you're breaking that phosphate bond on ATP, but you're also actually getting the phosphate and it's being moved on to a molecule to induce a conformational change. Any questions about enzymes? Again, this should all be review for you guys. Anyone taking biochemistry? You taking it now?
I think like biochemistry.
This is like baby biochemistry, or it should be. You taking biochemistry now? Okay. Anyone taking it now? You guys taking it here? Is it here though? They're offering it here though online? Okay.
Yeah, through STC, yeah.
Okay. Yeah, you guys, if you have trouble getting the class here at USF, David Merkler teaches it at USF. Really good. It's really hard, but it's good.
Thanks.
Okay. All right, let's keep going here for a little bit and then we'll take a little break. So the next section is on membranes. All right, so membranes are really important. Membranes are not just, structures that segregate things in the cell, although that's an important function as well, right? When you look at mitochondria, right? So they call the mitochondria the battery of the cell, and it actually functions a lot like a battery, the way that electrons move and things like that. But you've got two membranes that make up mitochondria. You've got an outer and an inner membrane, and it actually produces areas that not only differ in pH, but differ in the amount of protons that are in each section, in each area of the mitochondria. That's what we're going to get into it more later on, but it kind of makes it function like a battery. So these membranes are really important in creating compartments in the cell where you can segregate things, but that's not the only job of it. It has a mosaic of proteins that are embedded in them that provide very important functions. So when you look at membrane composition, it's about 50% lipid and 50% protein. And those proteins that I'm talking about are those proteins that are embedded in it that have diverse functions. And then half of the lipid content is cholesterol. So when you guys hear good and bad cholesterol, this is the good cholesterol. And what's the other half? I didn't put that on here, but you should know. It's 50% cholesterol. What is the other 50% of the lipid composition that makes up membranes? Phospholipids. Okay, membranes are made of phospholipids, cholesterol, which helps strengthen the membrane, and then you got all these proteins that are embedded in it. All right, we know the phospholipids. Remember from last time, we talked about this already. They are amphipathic molecules. They have a polar hydrophilic head, and they have nonpolar hydrophobic tails. The tails cluster together, so the heads are on the outside. And the membrane is very fluid. These phospholipids can move laterally. They can also rotate, move around, all kind of moving around. But what they can't do is move from one leaflet to the other. Right? So remember, the lipid bilayer is 2 layers, and we call each of those leaflets. And in order to move a phospholipid from one leaflet to enough, you have to use an enzyme to do that. It has a zany name called flippase, and we're going to talk more about that later on. But right now, just understand that these phospholipids can move laterally and they can rotate. And then you have, 50% of this lipid content is cholesterol, and it's embedded in between the phospholipids so that the polar hydroxyl portion of cholesterol is up there with the polar heads of the phospholipids, and it gives the membrane strength, right? Remember, these are just hydrophobic and hydrophilic interactions that are keeping membranes together. It's not very strong. So this cholesterol acts like a molecular glue to kind of give it some tension and some strength. Yeah.
Phospholipids can rotate and move laterally. What's the purpose of them moving laterally?
Well, I wouldn't say that there's a purpose, all right, but it does happen and it does have effects, right? And the reason it does happen is because there's no covalent bonds here.
Okay.
It's just like a soup of molecules that are stuck in that hydrophilic and hydrophobic orientation. But the effects that it has is it enables masses of proteins in the membrane to move together. And what ends up forming are something called lipid rafts. And these are where, these are like aggregated regions of particular proteins. Like when you look on cell surfaces, you don't have insulin receptors just evenly distributed around the cell, even though you would think that it would do that. They aggregate in particular areas. And it's like the fluid nature of these phospholipids that enables things like that to occur. And we're going to talk more about that later on too. Good question. So they call this a fluid mosaic model. Fluid because everything's moving. Mosaic because there's a lot of different components of the membranes, a lot of different proteins that are embedded in it. So a little bit about those proteins. We have membrane proteins that span the entire membrane, both leaflets, and they may have portions of the protein that extrude on both sides, right? Extracellular and intracellular. We call these transmembrane proteins. And there are the portion that is actually spanning the membrane are either alpha helical regions, or you can have beta barrels. So a beta sheet on its own will not span the membrane, but beta sheets can orient themselves into this structure, which is called a beta barrel, and a beta barrel can span the membrane. Right, so this is a good example of a transmembrane protein because it's spanning the membrane, and you've got regions that are found, extruding out of the membrane on both sides. All right, so a beta sheet can't on its own, but an alpha helix can on its own. Okay, so we see this most commonly in eukaryotic membranes, and the beta barrels are more common in bacterial membranes. Okay, so those are proteins that span the membrane. The other type of membrane-bound protein is a peripheral protein, and these proteins do not span the membrane. In fact, they're really not covalently bound to the cell. They just interact with other transmembrane proteins. And then you can also have lipid-anchored proteins. So these actually have lipid moieties that are added to them that anchor into the membrane. All right, so you got your protein, then you got a little lipid group on it, and it's the lipid group that sticks into the phospholipid bilayer because it's lipid, right? It can associate with the phospholipids and bind in there. The most common ones are it's a myristylation reaction that adds a myristatic acid group to it, or myristyl, it's called. You have palmitic groups or other lipid groups that can be added, and then also prenyl groups. So prenyl groups include, the most common one is farnesyl. And again, these are all lipid moieties that can add on to proteins to help them embed in the membrane. And then GPI anchors as well, right? So these are glycophosphatidyl'inositol groups, right? So it's basically sugar groups that add on to the proteins. GPI anchors. We're going to talk a lot about GPI anchors. All right, so you can see how this is a mosaic, right? You've got a bunch of different components that are embedded in these membranes. We've got integral membrane proteins, we've got transmembrane proteins, we've got peripheral proteins here, right? Here we've got a protein with a prenyl group, little lipid buoyancy that allows it to stick into the membrane. Right? Remember, alpha helices are the most common ones in eukaryotes that enable that transmembrane spanning. And then the beta barrels are more common in prokaryotes. We also see them in mitochondrial proteins because the mitochondria are prokaryotic, or they come from a prokaryote. All right, so there was an old saying, not an old saying, but old belief that like the guys that run the casinos are the valet drivers or the valet people, right? Why do you think that is? I don't know if that's necessarily true, but they know everybody that's coming in and out of the casino, right? So, the membrane is a structure that decides what gets in and out of the cell as well. So it's very important in defining the characteristics of the cell. We say that it's selectively permeable. It selects what gets in and what gets out. Permeable means to pass through. When substances diffuse, this should be very review for you guys, right? They move according to their concentration gradient. Natural diffusion dictates that substances will move from an area of high concentration to low concentration. So if I open up a bottle of perfume up here, right? Then I open it up and the alkene molecules that are dissipating off, they're in high concentration here, right? But they're going to move all around the room and span all around to where they're a low concentration. They're going to continue to do that until a state of equilibrium is reached. Okay. We can increase the speed of that diffusion by warming the room up. But, by this time tomorrow, if there's no air fluctuations in there, the concentration of those molecules should be the same all throughout the room. And that's what diffusion is, specifically passive diffusion, right? So no energy is required to do that, even though energy may enhance the rate of diffusion. So there are some molecules that can pass freely through the membrane, and these are small, uncharged molecules. Oxygen, carbon dioxide, nitrogen, water, and glycerol, and booze, and freely move through a membrane. Okay. Also, molecules that are hydrophobic. Now, hydrophobic molecules don't move very quickly across membranes, but they do move across membranes. When you think, a good example of this is the endocrine system, right? What does the endocrine system do? Produces hormones, right? So we got two classes of hormones. You got protein hormones and steroid hormones, right? So steroid hormones are highly hydrophobic because they are made from cholesterol, which is hydrophobic. And they will pass through the membrane. Protein hormones cannot pass through the membrane. They have to bind to surface receptors. So when you look at like the effects, the temporal effects that protein and non-protein hormones have, non-protein hormones react more slowly. It takes more time for their effect to get induced, but they're more longer lasting, right? Because they soak up into the cell, they wait around there. It takes time for them to diffuse out. Protein hormones, though, are really quick because they bind to a receptor right on the surface, and then they induce some sort of signal that quickly gets into the cell. All right. So those large polar proteins and molecules like glucose and things like that can't pass through the membrane. Also, charged molecules, which is protons, charged sodium, chlorine, calcium, can't travel through membranes as well. The membranes will, like I was saying with the mitochondria, I mean, it's a hydrogen house, right? There's a lot of protons that are moving around through the membranes within the mitochondria. It makes a very acidic environment in there. And they can't get out through, they can't pass through the membrane to get out. Right? So This is important. Be sure that you understand this and how the membrane is selectively permeable and what it's selectively permeable to, because there's a lot of ways I can ask exam questions about this. So it's important that you understand this very well. So for those molecules that can't pass through the membrane, you have to assist it. Typically, you have to use energy and you have to use a transporter to do that, right? And I kind of misspoke there. You don't have to use energy. It just depends on the situation. It also depends on the type of transport protein that you're using. So let's talk about transport proteins. You can have channel proteins. Channels are just pores, right? So they allow the passage of certain sized compounds to enter or exit the cell. Oftentimes they can open or close in response to signals from somewhere else in the cell. All right, carrier proteins are a little more complex. They're not just simple openings. They will usually change conformation, so they'll change shape to permit the binding of the molecule it's trying to move on one side of the membrane, and then it'll change shape to let it out on the other side. All right, so here you got a channel protein on the left there. It's really just a hole, but it's still selective, right? So only a certain size thing can get through here. Carrier proteins, though, are a little different. So they can actually undergo what's called conformational change, which I said earlier is when the molecule changes shape. You can see here when it's in its initial shape, it can receive the molecule that's being transported. And then once that binds, it induces a conformational change so that the molecule being transported can now exit on the other side. Has energy been used here? I mean, aside from the energy just from thermodynamics, like heat energy, no ATP has been used. So that means that these substances are moving according to the concentration gradient. They're moving from an area of high concentration to low concentration. They are diffusing. Whenever you have to move molecules against the concentration gradient, you have to use energy to do that. If you're moving things uphill, you got to pedal. And that is what active transport is. It requires the hydrolysis of ATP. You got to go from ATP to ADP. You got to get that phosphate. You got to get that free energy to move things across against the concentration gradient. All right, a good example, this is the proton pump. Proton pumps are found in the mitochondria, big time in the mitochondria, because they have to move protons to produce the gradient, which allows the production of more ATP. But it also involves conformational changes, and let's just go to the figure here, it'll make more sense. You can see, so protons here are moving against the gradient, right? So they're low on extracellular, which is, so they're low extracellular, they're high intracellular, right? So they don't want to move that way. This active transporter enables it to be done. So the proton can bind, ATP gets hydrolyzed, the phosphate gets transferred, some free energy gets transferred, and now the pump undergoes a conformational change and it spits the protein out against the concentration gradient, against the laws of nature. All right, you guys take a little break and we'll get on to the DNA stuff. Any questions or anything? If it seems like I'm going a little too fast, guys, just understand this, like this is all review. When we get to the new stuff, I will slow down a little bit, but what is it? Molecular science? Well, cell bio is a step to molecular biology. So yeah, I mean this is like baby molecular biology. Yes, sir.
The, what's it called? The carrier proteins?
Yeah.
Why wouldn't it cost energy to undergo conformational change.
So the ATP and the.
Because remember we were talking about substrates and enzymes and the allosteric site. When something binds to an allosteric site, that doesn't require energy to change the shape of it. Think of it like a monkey jumps on your back, right? It's going to change your shape, right?
Yeah.
It's the same thing. So when you're sticking things onto molecules, it's a stress to the molecule.
Oh,
It's a
but
strain.
you go through what reverse,
So this,
take it off, then it would cost energy.
not necessarily, no.
Yeah, I would.
So this right here, like this thing's just. God damn it. Just fitting in that pocket is enough to like strain them. Remember, when we're talking about energy, yeah, there's free energy going on here, right? There's this thermal energy that's working and moving molecules. But I'm talking about like ATP, specific energy. Adding chemical energy into the system is not required here.
Okay, it's already there.
Yeah, there's free energy there, but don't think of this as an energy driven reaction per se. Like, I mean, it's just diffusion. Energy is active transport in ATP. Just sticking that thing in there is enough to change the shape of the
Oh, really?
molecule, just like the monkey on your back or the allosteric inhibitor binding to the allosteric site of the enzyme changes the shape of the enzyme.
Okay, and
You're
that
not-
doesn't cost any energy.
you're not adding any energy to the system there.
Okay.
Okay. You're thinking like a physicist. Don't think like a physicist with this stuff because
Okay.
it'll mess you up sometimes.
So I just got it now. And they feel I get home, so I have my. No, you're good. Okay, the first thing I was looking for, I was like, okay, remember to ask. And then we're good. No, you're good. But my handwriting, this handwriting, I just started doing it. I'm like, you're out of the. And I also do my words, finding everything. Oh, so when I see those, I'm like, so. It's a one, so there's an I, it's an L. You're gonna, it's a number, yeah. Just. Yeah. Yeah, 14 valid. Just things round over. Okay. Not going to worry about it. Okay. Okay. So I would say, I feel like it was time over the now is when it's sometime it's very well. Yeah. So. Oh, those are one. Yeah. Yeah, I said, it's a name. Which was. We'll let you know. How would they see?
Hi guys, let's keep rolling here.
Yeah, like.
All right, we're on to a new chapter now. DNA, DNA. So this chapter is fundamentals of molecular biology. So we're going to cover all the sections in this chapter. The last section of it really condensed. There's also going to be a lot of spillover with the lab. So I do kind of browse through it a little quickly, section 3.3. But, molecular biology is study of the molecules of the cell. Yes, ma'am. Yes, ma'am. Sorry. So if we have a quiz next week, talk about that. Yeah, if we have a quiz next week, quizzes will only cover information from the previous lecture. Only stuff I go over today. Yeah, and that's again the point of the quizzes. They're not very difficult. They're just kind of meant to make sure that you're keeping up on the information. I think you maybe should be getting the idea at this time. You can't study the night before for these exams, it's just too much material, right? Remember a big, and I think you're going to see when you take the practice test today that, your success in this course is really also not only going to be heavily dependent on your ability to understand the mechanisms that we're going to learn, but also the language involved, right? So, the more aggressively you're studying this, that's what science is, like learn a new language. And you guys got to learn the terms and all that stuff in order to understand the questions. So a lot of reading, a lot of studying, a lot of review. All right. Yes. So quizzes will only cover was gone over in the previous class. All right. So this chapter is Introduction to Molecular Biology. The whole course, in my opinion, is an introduction into molecular biology. So the first section focuses on DNA, genes, and traits. This is what we stress in the field of genetics, right? Genes and traits. Genes are the basic units of heredity, right? It's a segment of DNA that codes either for a protein or an RNA, right? So if you have the gene for blue eyes, right? How many copies of that gene do you have in each of your cells? 2, very good. You got 1 copy from mom and 1 copy from dad. So maybe 1 copy is the gene for blue eyes, and then the other copy is the gene for green eyes. So those are alleles. They are variants of the gene. And then you being able to see that blue color is the phenotype. It's the visible characteristic. Although I like the term observable because blood pressure, things like that, those are all phenotypes as well. I guess you can see blood pressure in a person. Okay, so because you have 2 copies of it, if they're identical, if you have 2 copies of the blue eye color gene, then we say they are homozygous. But if you've got 1 blue and 1 green, we say they are heterozygous. And the way that genetics works is one of those alleles usually is more powerful than the other. It's dominant. And that's the one that actually carries through in the phenotype. So eye color doesn't work like that, but it's more complicated than that. But so the one that doesn't carry through to the phenotype is the recessive allele. The dominant, recessive, and it's how the alleles determine phenotype. That's really all we're going to do with that. You want more of that? Take genetics. Okay. But where does all that stuff come from? It comes from the chromosomes, right? The chromosomes, you have 2 copies of a set of chromosomes. This is for human genetics. And that makes the cell diploid. 1 copy you got from mom, 1 copy you got from dad. Where those copies came from were the gametes, right? The sex cells, the sperm and the egg, and each of those have only 1 copy of the genetic blueprint, so they are haploid, right? They are 1N. And the process to produce those gametes is known as meiosis, right? So meiosis is a variation of mitosis, which is normal cell division. So cells that are 2N, when they're mitotically active, make cells that are also 2N. So meiosis is like a variation of that. All right, if we change the DNA sequence to, we can potentially mutate, or we did mutate the DNA, but we could potentially alter the phenotype. Right? So mutations are both good and bad, right? Perhaps a mutation makes you more fit for your environment. Well, that's good. So it'll stick around for you and your progeny and make you more successful. But mutations can be deleterious as well. All right, perhaps it hyperactivates P53 protein. Well, then you're good chance you're going to develop carcinoma. All right, so this is like a hypothetical organism because it's just got 4 chromosomes, but you can see the diploid cell has 2 copies of each chromosome. And the male parent cells are 2N and then undergo meiosis to produce a 1N cell, which is the gamete, sperm, and the egg. and they come together during fertilization to produce a diploid zygote, which is called. Okay. And then that zygote develops into an embryo. So this is not completely accurate. But that's it. All right. So how did DNA actually get identified as being the informational molecule of the cell? Well, Griffith didn't do it, but his experiment was necessary for the next group, Avery McLeod, to be able to identify DNA as the magic molecule. But what he did is experiments with mice using a strain of bacteria called Strep pneumoniae. Okay, so Strep pneumoniae, he had two variations of this bacteria. He had an S strain, which the S stands for smooth because the colonies look smooth when they grow on the plate. And then he had an R strain, which the colonies, when they grow on the plate, look rough. Okay, the S strain was virulent, which means it killed the mice. The R strain, even though it was active Strep pneumoniae, was non-virulent. It wouldn't kill the mice. Okay, so he gave mice S strain, knocked them out. He gave mice R strain, it knocked them out. And then what he did is he heat killed the virulent strain. And when you heat kill it, you just heat it, you boil it. It doesn't completely destroy the bacterium, but you denature the enzymes to the point where they can't renature. You mess it up so bad that it's not a virulent strain anymore. So he killed S, and he mixed it with the non-virulent strain. And something happened. It still ended up killing the mice. So something transformed the smooth strain to restore its virulent capability. Okay, and this became what's known as the transformation principle. And you can easily transform bacteria, as we know today, by mixing bacteria with DNA and just heating them a little bit. The DNA will enter the bacteria. So you didn't know what it was in the R strain that was moving over to the S strain, but something was certainly moving. Okay, so just to wrap this thing up again, so the R strain is non-virulent. You give it to the mouse, mouse lives. The S strain is virulent. You give it to the mouse dies. You heat kill the S strain. You give it to the mouse lives. Right, but now you mix the heat killed S with the non-virulent R, and the S strain ends up killing it. Okay, so something is moving here from one bacterium to the next. Something was transforming when he mixed these bacteria together. But they didn't know what it was. So Avery, McLeod, and McCarthy basically repeated Griffith's experiment with some modifications. He took that R strain, and they did some purifications to them. They were able to purify protein from it. All right, they were able to purify DNA from it and purify RNA from it. So they purified all three of these things, and they found that when the DNA was added to the heat-killed S strain, it restored the ability of the S strain to kill the mice. Okay, so the big enzyme that they worked with, which made them famous, was DNase, right? So they also worked with some proteases to break down the proteins of the DNA, so they only had the other molecule left over. All right, this clearly established DNA as the genetic material. Okay, so here you've got smooth and rough. And what he did is he added the purified sDNA to the R strain of bacterium, and it restored the ability to be virulent. All right, so just knowing that DNA is the magic molecule is not enough, though. Like, to determine how something works, you really need to know how it's built. You need to know the structure of it. So the guys that did that, of course, are Watson and Crick. In the 1950s, they published their famous paper. And you guys should read that paper. It's only like 2 pages long. But it is credited with being one of the most prominent scientific reports in history, you know, right up there with Newton's thing and Einstein's stuff. Problem with it though is they didn't get permission from someone to use their data. Big no-no. But Rosalind Franklin was a woman that did some cutting edge work with X-ray crystallography. Okay, so if you want to study what a molecule looks like, a molecule that's invisible, as all molecules are, what you do is you make it bigger. So to make it bigger, you make a crystal of it, right? So it crystallizes and it grows into the same shape that it originally is in. You know, not big enough still that you can see, but big enough that you could actually work with it to the point where if you bombard it with X-rays, the X-rays will deflect off of the molecule, and then you can capture the patterns of that deflection and, you know, assemble a model of what the molecule actually is structured as. This is called X-ray crystallography and X-ray diffraction. And she did the work that determined, you know, that the double helix of DNA. There was another guy, Erwin Chargaff, who determined that the amount of A that you have, the adenines always equals the amount of thymines, and also the amount of guanine that you had in the reaction always equal the amount of cytosine. So Watson and Crick actually didn't do a single experiment. They used all of this data from Chargaff and Franklin to assemble the double helix model and also the complementary base pairing that existed within the molecule. Okay, so DNA has complementary base pairing, A's to T's, C's to G's. It has polarity, right? So you have the phosphates and the hydroxyls that are making, you know, that polarity, 5' to 3'. It's double-stranded, and it can be replicated accurately in a semi-conservative manner, which means that the molecule can separate, and one strand of it can be used as a template to build a whole new molecule of DNA. All right, this is what the X-ray crystallography things would look like. Trying to interpret that without a computer, I can imagine, is a nightmare. You know, the double helical winding staircase shape of DNA gives it some very important shapes. So it gives it these grooves, major and minor grooves, which we'll see are very essential later on for the binding of DNA binding proteins that will latch on to DNA and whatnot. All right, so these guys, Messelson and Stahl, used radioactive nitrogen. Why would they use nitrogen? Yeah, so nitrogen is kind of a unique component of DNA. You know, you don't have any in carbohydrates. There's some in proteins as well, but using that, you can radiolabel DNA. So he would grow it in this heavy N15, and then he would shift the culture over to a lighter N14 medium. And then he could analyze the DNA by using cesium chloride gradients, all right, which is basically a tube of cesium chloride. You spin it at a really high speed, and even small amounts of DNA you can separate out in this column. So you ended up getting like an intermediate band between 14 and 15. So that means that half of the molecule is the heavy N15 and half of it is N14. And it proved that, you know, DNA is definitely replicating in a semi-conservative manner, where half of the molecule is conserved from the parent strand and half of the molecule is new. Right? So, yeah, so double-stranded molecule, you separate it out. Here are the two old strands, and DNA polymerase comes in and it builds a complementary strand to each. So, you know, half of the new strand is old, half of the new strand is new. Right? This was proven by cesium chloride density centrifugation. So you have your light DNA and your heavy DNA, right? And then you have this intermediate hybrid DNA, which represents the, you know, 50% of the daughter strand being new and 50% of the daughter strand being old. Okay, so there's DNA. So DNA, though, is just a molecular message. It's a code of A's, T's, G's, and C's that somehow can provide information on how to work this and define a cell. And that happens as a result of the central dogma of the cell, right? Going from DNA to RNA to protein. Going from DNA to RNA is transcription, and going from RNA to protein is translation. So, you're not modifying the molecule, you're building a new molecule. So you're reading DNA and creating an RNA molecule that is essentially a complement of one of those strands of the DNA. And you're creating a protein in translation that is based on that RNA sequence. This is the flow of genetic information. Now, there's a few different types of RNAs in the cell. All right, we've got the three we're going to talk about in this section are the mRNAs, the rRNAs, and the tRNAs. There's a lot of other like microRNAs, picoRNAs, and things we'll talk about later on. But all of these three types of RNA kind of converge at transcription and translation, even more so at translation. The type of RNA that gets made as a result of transcription is mRNA or messenger RNA. And messenger RNA because it carries the genetic instructions from DNA. Then that messenger RNA needs to travel, after it's been transcribed, needs to travel to a ribosome to get translated. Well, the ribosome is made up of about 50% rRNA. It's called ribosomal RNA. The ribozymes we were talking about early on, rRNA is a ribozyme. It's intrinsically catalytic. It can actually produce the peptide bonds that are involved in doing translation. And then finally, the tRNA is an adapter molecule that kind of brings the amino acids into this process of translation. And it brings it in the right manner so that you produce an amino acid code that's based on the RNA code. So that mRNA is where we're starting from. It's the template for translation. All right, so here is a strand of DNA that's been separated. And one strand of the molecule is read by the enzyme RNA polymerase. Polymerase because it's polymerizing a piece of RNA. Right? It's reading the DNA and it's building an RNA molecule here. And you can see, remember, RNA doesn't have T's, it has U's. So wherever there would be a T, because there's an opposing A, have a U put in instead. All right, something to note here, which moving forward is going to be very important. Might as well memorize it now. When these nucleic acids are polymerizing, they always add new nucleotides to the 3' end of the molecule. The only way these things grow, DNA or RNA, everything is always being added to the 3' end. So new nucleotides are coming in. There's the 5' end of the RNA. Here's the 3' end of the RNA. That means here's the 5' end of the DNA. Up there is the 3' end of the DNA. Okay, because it's anti-parallel. Just like DNA to DNA is anti-parallel, DNA to RNA is also anti-parallel as well. There's a repeat slide there. Yep. All right, here's the third RNA molecule, the tRNA, which behaves as an adapter molecule. Okay, now how many amino acids are there? 20. Right? For every amino acid, there is a tRNA. Okay, and for every tRNA, there is an enzyme called RNA synthetase that catalyzes the addition of the amino acid to the corresponding tRNA. Okay, so 20 amino acids, 20 different tRNAs, and 20 different enzymes that catalyze the addition of the amino acid to the tRNA. Right? So tRNA is an RNA. It's ribonucleic acid that's folded up. Remember these, so what's kind of unique about RNA molecules. They can fold up into shapes that permits their function. And these tRNAs have arms that extend out. Another one that kind of extends down. And there are 3 nucleotides that project out on the bottom of the molecule. We call it the anticodon area. Then up here is where the amino acid gets covalently bound by the tRNA synthetase. Okay, so this one is adding the histidine to the tRNA. We call it charging as well. Okay, we say the tRNA is getting charged by adding the correct histidine on it. So the cell is a soup of all of these, the cytoplasm, you have the soup of all these charged tRNA molecules that are available for the ribosome to do translation. So they can come into the ribosome, whenever needed, so that you can add the correct amino acid into the sequence. All right, so that anticodon region, is unique for its particular tRNA. And you can see it binds, or it's complementary to a sequence on the mRNA. That's how the code is able to go from RNA to protein. Because this is one codon, here's the correct tRNA for that codon, because it has the corresponding anticodon. And then the next tRNA can come in and bind to the next codon region, providing maybe the same or a different amino acid, depending on the codon of the anticodon. Yes, ma'am.
Question.
Sure.
At the bottom, I don't understand the part where there are only 3 codes.
Well, so this whole thing is there's hundreds of nucleotides that make up this RNA, but there are 3 that are found in this area of the tRNA
Right.
that make up what's called the anticodon, because they project out and they can hydrogen bond to a corresponding codon, which is found on the mRNA. Let's see if I can find a figure that kind of shows it a little better. It's not yet. And this is the best I've got right now. So, but this is all happening like in the ribosome during translation. You guys remember from Bio 1, we're going to talk about it later on. There's 3 pockets in the ribosome, right? So the tRNA, here's your mRNA, which is like a piece of tape. And then your tRNAs come into these pockets, and the anticodon binds to the codon of the mRNA. Okay, this is where the peptide bond is forming, but the charged tRNA is what's providing those amino acids for this process here. This is, remember, what is this called? Translation. If you're still not getting it, may want to take a break. I'll pull up a figure that'll show it a little better. All right, so codon, anticodon. If you guys remember Bio 1, codon is 3 adjacent nucleotides, right? And they code for an amino acid. Yeah. So it's a triplet code, and there were some very prominent experiments early on that figured out why it was a triplet code, how it could be a triplet code. So if you have a long sequence of nucleotides, so let's identify the codons here. Right, so that is a reading frame. Each of those codons corresponds to a particular amino acid. And in order to figure out what the amino acid is that it corresponds to, you would use what's called the codon usage chart. Okay, so ATG, I know, is the start codon, but this is the first letter on the left. Okay, so A, and then the second letter. So this is mRNA, so AUG. So A, U, and then G is over here. So this corresponds to methionine. Okay. And then the next one would be CCG. So C, C, and G. So the next one would be a proline. And so on. Okay. Now, it's known that it's a triplet code, that's the reading frame, because if we add or remove one or two of these nucleotides, it completely messes up the reading frame. So let's do that. Let's make it A, T, G, and remove these two C's. Now we do G, A, T, C, C, G, A, T, C, A. Now our codons are completely different. Okay. So this is called a frame shift mutation. When you add or remove two adjacent nucleotides, it shifts the reading frame. And that's the early experiments that were done to identify it as a triplet code involved that. Okay. There were experiments that involved this viral gene. And you can see here, like your DNA code has particular amino acid code that corresponds to it. But if you change, okay, I'm sorry, if you remove two of the, add or remove two of the adjacent nucleotides, now you completely mess up the reading frame and you change the sequence of the amino acids. Okay. Now, if you were to add or remove three, well, that would add or remove one amino acid. Maybe that'll completely mess up the protein. Who knows? But this was how the experimental evidence was derived to determine that it was a triplet code. All right, Nierenberg and Mathai, these are the guys that basically discovered translation, and they did it by doing in vitro systems. So they would take what's called a rabbit reticulocyte, and those contain all of the necessary elements to do translation. It contains ribosomes, it contains charged tRNAs, and then you introduce an mRNA, and you can actually produce a protein in the tube of that system. So they messed around with that, and they used these polynucleotides to do that. So they would have mRNAs that were just a string of U's, or a string of A's, or a string of G's, and so forth. And they would see that that would make a particular sequence of proteins. And they did that over and over again, and they basically discerned the whole protein and codon code. Okay. So obviously this will be provided for you on a test if you need to work with the code and whatnot. Okay, so I've skipped over a couple slides here. Sorry about that. All right, so you've got 20 amino acids. You've got 4 nucleotides. Right, now we're talking about mRNA, so it's A, U, G, and C. So you've got 64 possible codons, though. Okay, so 3 of those don't code for an amino acid. They actually code for the stopping point of translation. So at the end of this mRNA, you're going to have what's called a stop codon, which tells translation to stop. You're going to stop building a protein. One of the codons that codes for an amino acid is the start codon, right? AUG. In DNA, it's ATG. But so that indicates where translation starts. So in eukaryotic systems, the first amino acid of all proteins is always a methionine. Now, in some proteins, that methionine will get cleaved off, but oftentimes it stays. But the first amino acid of all eukaryotic proteins is always methionine. Okay, so now let's look at this. So we have 61 codons that code for 20 amino acids. So does that make sense? All right, so what's happening here is something called redundancy, right? So there are multiple codons for the same amino acid. There is flexibility within the codon code to code for a particular amino acid. Look at leucine, for example, right? There are 4 different codons for leucine: CUU, CUC, CUA, and CUG. The first 2 positions of the codon are the same for all leucines. It's the 3rd position that has flexibility often in these individual codon sequences. So the 3rd position being flexible is what's called the Wobble hypothesis, because that last nucleotide can change and it doesn't necessarily affect what protein it is. That's not always the case, because some of these only have 2, right? Lysine 2, right? But a lot of the nonpolars have. Different ones, okay? All right. Okay, so DNA is read, mRNA is made, and we're going to start to label the different regions of these molecules according to how they kind of situated in the central dogma. Right? So there are regions that flank the portion of the mRNA molecule that's actually going to be used to determine what the sequence of the amino acids are, which is called an open reading frame, or ORF. There are regions that flank that are untranslated regions. So all this stuff has been transcribed, but the UTR is not going to get translated. Only the ORF is going to get translated. This is a good figure. So we've got a piece of DNA, and it undergoes transcription. RNA polymerase is the magic enzyme that does that. It reads one side of the DNA molecule. Remember that figure about 5 figures back? And it builds a piece of RNA. Okay, now that whole RNA molecule is not going to be read to become a protein. Right? There are regions that flank the open reading frame called untranslated regions, or UTRs. There's a 5' one and a 3' one. Yes, ma'am. They can be, but so there's important regions in there that are important for like regulating translation. So let's actually start learning this now. So for prokaryotes, there's a sequence in there called Shine-Dalgarno sequence. And in eukaryotes, there's a sequence called the Kozak sequence. And Kozak overlaps a little bit into the open reading frame. It contains the start codon, but both of these are like components of the UTRs. And then there are other sequences in the UTRs that will contribute to things in other ways, but it's an untranslated region. Actually, this was the figure I was looking for you earlier. So this shows the mRNA with the tRNAs coming in and the amino acid chain being built. All right, one thing to kind of latch onto a little bit here is the polarity. So RNA is 5' to 3'. And look, as the peptide is growing, the 5' end corresponds to the amino end of the growing peptide, and the 3' end of the mRNA corresponds to what? It's the other end, carboxy, right? Carboxyl end. Okay. So this is just a representation of the open reading frame. All right. So that is transcription and translation in a nutshell. We're going to learn those processes in detail, though, in later chapters, especially the translation's a big one. Some of these processes have, 20, 25 molecules that work together to kind of complete it. So it's going to get more complicated. All right, now the process going to DNA to RNA to protein, exists in all eukaryotic cells and prokaryotic cells, but there's a little modification of this that exists in nature that has been brought about by viruses. So viruses have a modified version of RNA polymerase that can actually do reverse transcription. Okay, can actually read RNA and create DNA. That enzyme is called reverse transcriptase. Reverse transcriptase reads RNA and builds a DNA molecule based on it. That DNA molecule has a special name. It's called cDNA. Right? It stands for complementary DNA. So cDNA is the type of DNA that's made by reverse transcriptase reading RNA molecules and building DNA. The viruses that do this are called retroviruses. And retroviruses are RNA viruses. Viruses like influenza and HIV, those are good examples of RNA viruses. And it's actually because of this reverse transcriptase, which is why they're so difficult to prevent, because they change, they mutate. Reverse transcriptase is not a very effective enzyme. That's the wrong word. It's not a, it doesn't have good fidelity. Okay. It makes a lot of mistakes when it's making that cDNA copy. So that mutates the enzyme. So here we've got viruses. This is a retrovirus coming into a cell. And the RNA, so the virus codes for and contains reverse transcriptase. It's an RNA virus. But in order to get integrated into the host chromosome, you can't integrate RNA into the host chromosome. So that essentially that RNA needs to become DNA. So reverse transcriptase will perform reverse transcription on the RNA, create a cDNA, and now that cDNA can become part of the host chromosome, and the host cell now can make multiple copies of the virus. Right? When that cDNA is integrated into the chromosome, we call it a provirus. It's part of the viral cycle. All right, let's see here. So you guys just take 2 minutes. I'm going to get through some of this chapter, and then we're going to, this last section, just the last section. And then we'll take our practice test.
You know, come back to it.
So what I do is while I'm here, such as live. And I go back to this video. And then I leave this and I take it up to the doctor's house. Being around confused, I go back to the house. And then if I'm still just saying, I feel.
Right what we're doing. Yeah, I'm thinking about like. The one I haven't been interested in. Where's working my. Like, even if I know it, I've got to do. Yeah, I have to have something to say that. Yeah. Well, I will, so thinking about the next thing where I'm going, yeah.
By the way, you guys did very well the other day in lab, so please keep that up the rest of the semester. I don't know if I already told you that today, but that went very smoothly.
Are we taking attendance?
I don't take attendance in lecture. I don't care if you show up. But I don't. So I don't. The only thing is, if you don't show up, you can miss a quiz, miss a. But you know, if you want to, go ahead.
The tests themselves are in the class as well?
Yeah.
You need to be here for that.
Those days are designated on the schedule. And unless there's a storm or something, those days won't change. Yeah, we'll do the test first though. So that you want to, but I'm gonna keep talking after the test. So we'll do material after the test, not too much, but whatever's put up on the schedule. But you can get lost if you want.
Get lost in me.
If you want, you guys are adults. I mean, I think the more often you hear the information, the better you're gonna learn it. So you know, unless you got something better to do. Yes, ma'am.
Names,
Dates, I'm not a big date guy. These are some of the more, you should be familiar with the experiments that were done and discoveries. Yeah. I mean, that's pretty much it for names for the semester. Yes, sir. Just real important, like, because like the movement of those molecules through the membrane, there's a lot of ways I can ask questions about that. So you really need to know it. Like, you gotta really have it memorized to, with the polarity and stuff and the hydrophobic, hydrophilic, all that stuff we covered last time. You can see you've got to really know that information to understand what you're reading and talking about moving forward.
Like the WX model.
Uh-huh.
Or,
Yeah.
we do.
Yeah, they're pretty famous. Watson and Crick. And,
Okay.
yeah, you should know this. But that's it. I'm not doing any. I don't think we have any more names going forward. Yeah, they're just like what you're going to take today, except longer. Today's just 10 questions. So I'm giving you, it's 40 questions and you've got an hour and 15 minutes. All right, guys, let's keep going here. I don't know if we'll get through this whole thing here, but if we don't, that's okay. Last section. All right, so the last section of this chapter is an introduction to the techniques and procedures that can be used to analyze DNA, analyze this all-important coded message that is invisible, right? So what sort of techniques can you use to study DNA? And it's kind of coined recombinant DNA technology. So if I think I've said this before, but you guys, if you watch like those pharmaceutical commercials and you see like rDNA, right? That stands for recombinant DNA. And that means that DNA has been modified to, produce a vaccine or produce some sort of compound. Did you have a question? Well, multiple choice. There's multiple choice. No. This is your name. Okay, so that's why we say rDNA, because you're manipulating DNA. So you're isolating it, you're sequencing it, you're actually determining the order of the nucleic acids, or you're manipulating it, you're cutting it, you're moving it to another piece of DNA, things like that. Okay, so this has enabled us to determine, genome sequence and how genes are built, how they're structured, where the domains code from, homology among different organisms, right? So if this gene is more or less similar to the same gene in another organism. All right, the most fundamental aspect of manipulating DNA, because chromosomes are so large, is the ability to cut the DNA into smaller portions so that you can manage them. And it's enzymes called restriction endonucleases that do that. Okay, so endo means within, nucleus means to digest or to cut. So these are enzymes that cut inside the DNA, right? For example, there is a prominent restriction enzyme called EcoR1. The sequence it cuts is GAT2C. Oh. GAT2C. Okay, so here's your double-stranded DNA molecule. And this enzyme will clamp down on this specific sequence. And when it cuts it, it'll cut the DNA molecule like this. Okay. So kind of like a little IQ test for you. What is unique about the sequence? Palindromic, very good. Right. So that means it's the same going forward as it is going backwards. It's like the word Bob. Right. And that's what these restriction enzymes recognize, are these palindromic sequences specifically that bind to and cut DNA at particular locations. So there are, yes, ma'am. EcoR1 is the enzyme that that's a restriction site, but I think AQR1 recognizes that site. GAATTC, I got it right. Okay. So there are hundreds of these commercially available, and they all originate from bacteria. Bacteria produce these enzymes to protect them against viral infections. All right, bacteria can be infected from viruses too. Those viruses are called bacteriophage or just phage. And, they are nucleic acid. That's what viruses are. So if you have an enzyme that can chop up that virus without hurting itself, without affecting its own chromosome, that's a great way to protect itself. But they are an invaluable tool in molecular biology because they can cut pieces of DNA at specific recognition sites. And how they leave the ends of the DNA after they cut it is important. So this situation here, you can see there's no nucleotides like overhanging one side of the sequence. There's no overhangs. Here, there are overhangs. Okay, so we call these type of enzymes blunt cutters. And we call these overhangs, we call them sticky ends, right? And the reason that's important is because these things can then re-anneal. They can come back together and form a complete DNA molecule again. And these can as well, but if you're trying to direct the synthesis of DNA in a particular order, you can use these sticky end sequences to do that more effectively, because this could stick to any other blunt end, but this can only stick to an end that's complementary to it. So you can use these enzymes to do something to actually map the genome, essentially, and we call this restriction mapping. So, using the correct enzymes within a sequence can produce a number of certain size DNA fragments that can be used to actually assemble that original sequence. In molecular biology, they call this a scaffold, right? So it's like it's these pieces of DNA of particular sizes that are produced from cutting up the genome. All right, let's look at an example. So you've got bacteriophage lambda DNA, right? This is like a well-studied piece of DNA. And you cut it into 6 fragments, okay? So these fragments range in size from 3.6 to 21.2 kilobases. A kilobase is 1,000 base pairs. Now, the way to evaluate the sizes of DNA is through agarose gel electrophoresis. Okay, we're going to do a lot of agarose gel electrophoresis. You guys loaded a gel the other day in class. Right, agarose separates DNA based on size, so that smaller fragments of DNA migrate more quickly, and larger fragments migrate more slowly. So the banding pattern is contingent on the size of the fragment. So our piece of DNA there is cut with EcoR1, and it produces all these different sizes of DNA that we can now see on an agarose gel, where the largest one, that 21, right there, is up here on the gel, and the smallest one, the 3.6, is down here. Remember, DNA has the negatively charged phosphate groups, which enable to migrate in an electric field, so it always travels to the positive electrode. And the larger fragments are retarded more by the agarose, which is that porous matrix, right? Has these little holes that the DNA can move through. All right, so this is called restriction mapping. You're mapping the DNA based on fragment sizes as a result of cutting it with an enzyme that cuts it at specific sequences. All right, so now you've cut your DNA. What are you going to do with it? Well, you need to put it into somewhat of a working system. And plasmid vectors make very good working systems to study DNA. There are also thousands of these commercially available. If you ever want to check them out, the company that does these is called Adgene. That's a good one. get plasmids from Adgene. The company for restriction enzymes is, it's my brain just went, you can do Promega for restriction enzymes as well. All right, so these companies commercially sell these things. Anyways, a plasmid vector is also called a cloning vector because you can put a piece of DNA in it and now you can work with it. We'll talk about working with it later on. But these plasmid vectors come from plasmids. Plasmids are extra chromosomal pieces of DNA that are found in bacteria. So here's your bacteria. Here's your bacterial chromosome, which is a single circular piece of DNA that's found in the nucleoid region. There's no membrane. But also found in bacteria are these smaller extra chromosomal pieces of DNA, which we call plasmids. The reason that we have these plasmids, but bacteria have them, is they code for antibiotic resistance. So all that stuff with resistance to antibiotics is because of these plasmids in them. They all have key features, right? All plasmids have, when you look at them, they're circular. You know, they're small in terms of like anywhere from 2 kb up to 100 kb. Usually given names like under lowercase p, which stands for plasmid, and then some name. They have to have a replication origin, which is called ORIC, right? Origin of replication, so that the bacteria can copy the plasmid. Bacteria, plasmids, because their role is usually in antibiotic resistance, will also have a resistance cassette. Right? This one makes the bacteria resistant against ampicillin, which is a family of antibiotics. So that gene codes for some enzyme that messes up ampicillin. Another key feature of plasmid vectors is what's called a multiple cloning site. Right? All those DNA fragments that we cut with the restriction enzymes and we want to move into our plasmid, we're going to put them in here in the multiple cloning site. It basically is a series of multiple unique restriction sites that we can put our gene that we're studying in. Okay. All right. Plasmid, extra chromosomal gene that confers antibiotic resistance. See, origin of replication, multiple cloning site, antibiotic resistance. Okay. So we got our gene, it's cut with the restriction enzyme. And we've got our plasmid that has the same restriction site in it. So here's our plasmid, here's the multiple cloning site, and in the multiple cloning site, there is an EcoR1 site. Here's our gene that we cut out of a piece of DNA, and it has EcoR1 sites that flank it. So now we can take this plasmid, we could also cut it with EcoR1. And now it's going to have flanking EcoR1 sites. We can take this and mix it together in a tube and add an enzyme called DNA ligase. That will produce the phosphodiester bond that will ligate this gene into our plasmid. Okay, we have just done basic molecular biology. We have shuttled a piece of DNA into a plasmid cloning vector, and now we can do things with the DNA. Okay, because there's a lot of different cloning vectors available that have all these different features. They might have different promoters that will drive expression of the gene. They might have bioluminescent tags that will make the gene glow in a certain cell. All different things, and we're going to talk about a lot of different examples of that throughout the course. Somebody's drilling up there. All right, because it has that origin of replication too, we can transform bacteria by putting that plasmid DNA in bacteria, and the bacteria will bulk up our DNA for us. All right, so this is showing you You're cutting your insert with EcoR1, you're cutting your plasmid with EcoR1, so they have complementary overhangs. That's why this presence of sticky ends makes those restriction enzymes a little more useful than the blunt end cutters. And then you can still use a blunt end cutter, but oftentimes you can't get it into the right orientation. But now we can add DNA ligase and fuse these things together and produce a continuous piece of DNA. So the general workflow with cloning is you digest DNA with a restriction enzyme, you digest the plasmid with the restriction enzyme, you mix the two cut pieces of DNA together and you ligate them together. Now you have a circular piece of DNA that has your gene in it, and then you transform that into bacteria, just like Avery McLeod did. And now you grow your bacteria and it grows up a lot of copies of our DNA. We can purify the DNA, we can take the DNA and put it to other cells and things like that. Ugh, that's a nasty looking figure. But okay, so just what I said is occurring here. You got your cloning vector, you cut it with EcoR1. Here we've got a pool of human DNA genes, right? A bunch of different genes. We mix it together in our tube, we add DNA ligase, so look, it starts to produce what's called a library of genes. We put it into bacteria and we grow it, and when they grow out, each colony is a clonal population, right? So each colony contains all the same bacteria, which means that all the same bacteria have the same gene in it, whatever it is. And that's why you can get on a plate representative libraries of genes. That's why they call this genetic library. Now, one thing about the genome is, you cut a piece of DNA with the restriction enzyme and, well, let's say this is our chromosome, which is millions of base pairs long. This is gene X, Y, Z, and P, and I'm selecting this marker. And we cut it with the restriction enzyme, and, unless you know the sequence, you don't know where it's going to cut. Maybe it cuts here and here and here and here. It's not cutting our genes out neatly. So there's a version of generating libraries that incorporates in reverse transcriptase. So with reverse transcriptase, you can take all of the messenger RNAs that are in a cell. Sorry, that means I should shut up. I'm going to go over this last slide because that's the whole lecture. You can take all the mRNA that's in a cell. Okay, so here's our cell. It's got all the mRNAs that have been transcribed from the genome. So the thousands of genes that make up that cell, there's an mRNA for each. You can purify it out of the cells, and then you can treat them with reverse transcriptase. What's that going to do to for each of those RNAs? Going to make a what? Going to make a cDNA copy of each of those transcripts. So now we actually have the sequence for the protein coding gene. This is useful. With the cDNA library, now you're actually studying the genes. The DNA is a more stable form of the RNA. That's why using reverse transcriptase is very good. But the problem with it is reverse transcriptase does introduce a lot of mistakes in the DNA version. But that has been, reverse transcriptase has been genetically engineered to have much better fidelity and work better in the lab. But here you've got your mRNA, you're making a cDNA copy. You then have to add a DNA polymerase to make double-stranded cDNA. And then there's a step where you add adapters onto the cDNA. You add these linker sequences that introduce restriction sites. You can now cut it with the enzyme. Now you can ligate that into your cloning vector. And now you have what's called a cDNA library instead of a genomic library, which is just, pieces of the genome. Might not be as relevant to study as a cDNA library. Usually the cDNA libraries are more useful, but it just depends what you're doing. Okay. All right. The last thing in this chapter actually is dideoxysequencing. I'm not going to go over that today. I will do that next time because that's a little involved. And that'll be it for today's lecture. So just take a minute and then we'll do our practice exam and then you guys are out of here. And honestly, if you want to leave now, you can. You don't have to take the practice exam. You didn't have to be here at all. It's up to you. Stop sharing. Can actually.
If you